What is the cell cycle? So, hey y'all, my name is Yaj, and I work with Roman and Kale on this project. So, I'm going to be telling you guys all about the cell cycle and how a single cell divides into two separate ones. So, this process is fundamental to life, allowing organisms to grow, repair damaged tissues, and reproduce. So, without further ado, let's dive into the cell cycle. So to understand how a single cell divides into two, we first got to learn some basic terminology. So that being said, let's talk about chromatin. So chromatin is a term used to describe DNA that's wrapped around histone proteins. So during cell division, this chromatin condenses into more organized structures called chromatids. To make an explanation simple, we'll use an imaginary cell with 6 chromosomes, or 2n equals to 6. So 2n indicates this is a diploid cell, meaning it has two sets of chromosomes, one inherited from each parent. The number n represents the unique sets of chromosomes within each cell. So keep this in mind as we move forward. So the next step is where most of a cell's life is spent. The phase is called the inner phase. So this is where the cell grows, prepares for division, and it performs normal functions. The inner phase has three stages: G1, S, and G2. So breaking these down, first up we have the G1 phase. Or the gap phase. So during this period, the cell undergoes rapid growth. It synthesizes proteins and produces organelles, which are essential components for its proper function. This phase is all about preparation. And then next up is the S phase, which stands for synthesis. This is where the magic happens. The DNA gets duplicated. By the end of this phase, the cell has two complete sets of genetic material. Um, while the amount of DNA doubles, the number of chromosomes stays the same because of the duplicated DNA strands remaining attached to the centrome. And then finally, we have the G2, or the second gap phase. In this stage, the cell continues its preparation for division by synthesizing additional organelles and molecules needed for the process. By the end of G2, the cell is fully equipped and ready to enter the next phase of the cycle. So the next phase is the M phase, also known as the meiotic stage. So this is where the actual cell division takes place, and then it consists of two main processes: mitosis, which is where the nucleus divides, and cytokinesis, where the cytoplasm splits. So let's break down mitosis. It has four stages: prophase, metaphase, anaphase, and telophase. So remember PMAT. So for prophase, the chromatin condenses into visible sister chromatids. The nuclear membrane dissolves, and spindle fibers start to form. These spindle fibers originate from structures called centrosomes, which move to opposite ends of the cell. This sets the stage for chromosome movement. And then, moving on to the second stage would be the metaphase. So, during the metaphase, um, chromosomes align at the center of the cell, forming what's called the metaphase plate. Uh, Spindle fibers attach to the centromeres of each chromosome, and they ensure they're ready for accurate separation. And that's basically all for this phase. Meiotic, yeah. Mm-hmm. And then the third phase is called the anaphase. So in this phase, the sister chromatids are pulled apart by the spindle fibers and moved toward opposite ends of the cell. At this moment, the chromosome count temporarily doubles, ensuring that each new cell will have the correct number of chromosomes. And then, after that lengthy process is over, we finally can move on to the telophase. So, the telophase has uh, the separated chromatids reaching opposite poles of the cell. Uh, New nuclear membranes form around each set of chromosomes, and the spindle fibers disassemble. At this stage, mitosis is complete, but the cell itself isn't finished. And nearing the ending of mitosis is when the cell actually begins to pinch in two, which is going to be leading into our next thing, our final step, which is going to be called cytokinesis. So, in cytokinesis, the cytoplasm divides to form two distinct cells. In animal cells, uh, this happens when the membrane pinches inward, creating a cleavage furrow, and in plant cells, cell plate that forms between the two. Uh, and then the last phase is the GO phase. So now, not all cells continuously go through the cycle. Some enter a resting phase called GO. So in GO, rather than focusing on division, they exit the cycle and they much rather focus on survival. So examples would include nerve cells, which typically remain in this state unless reactivated under specific conditions. So so it's more on just surviving and not necessarily needing to duplicate. And then yeah, that's basically the cell cycle. That's the end. Thank you. I hope you all enjoyed.